Hello, everyone, and welcome to today's webinar. FED-AMR, the role of free extracellular DNA in dissemination of antimicrobial resistance over ecosystem boundaries along the food feed chain. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by OpGen. To learn more, visit opgen.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. All questions will be answered via email address after the session today. I'd like to now welcome our speaker, Werner Ruppitsch, PhD, tenured researcher, Institute of Medical Microbiology and Hygiene, Division for Public Health, a part of the Austrian Agency for Health and Food Safety. Dr. Ruppitsch, you may now begin your presentation.
Thank you very much for the kind introduction and the invitation to present the project about the role of free extracellular DNA in the dissemination of antimicrobial resistances in an agricultural environment. RS Genetics was an external partner in these projects. We have no other conflicts of interest to disclose here. The project was funded within the Horizon 2020 One Health European joint program with 2.4 million euros, and the consortium consists of 12 contributing partners. The project will end by the end of this year, and it is a multidisciplinary approach including analytical chemistry, microbiology, genomics, epidemiology, bioinformatics, and statistics. The background of this project is the fact that the contamination of the environment with antimicrobial resistance genes is considered a global threat to the environment itself, but also finally to public health. And WHO recognizes antimicrobial resistance as one of the critical challenges of public health in the 21st century. If we have a look into research and into literature, we find out that most of the environmental studies dealing with antimicrobial resistance genes are based on total DNA. So the role of extracellular DNA is still neglected in most of the environmental studies. And this limited knowledge that we have about the role of extracellular DNA in the environment has also to do with the challenges to isolate extracellular DNA of good quality and also in high enough amounts from environmental samples. Per definition, extracellular DNA is all the DNA that we can find outside living cells. It can persist for quite a long time in the environment. What we know from the literature, from 21,000 years to 900,000 years, depending on the type of environment. The size of extracellular DNA ranges from 8 base pair to 20,000 base pair. The abundance in comparison to intracellular DNA is quite low, with 100 to 1,000 copies of antimicrobial resistance genes or mobile genetic elements per milliliter of wastewater. Therefore, natural transformation is not very likely for extracellular DNA, but due to its long persistence, hotspots can be performed in the environment which increases then the probability of horizontal gene transfer. Extracellular DNA is also an important structural component of biofilms, and in these biofilms, extracellular DNA contributes to the dissemination of genetic information and finally also contributes to the transfer of antimicrobial resistance in biofilms and antimicrobial resistance of biofilms itself. We also know from research that extracellular DNA is used in plants and also in human body to inhibit the growth of bacterial cells and also kill bacterial cells, as you can see on the figure on the right side. It's also important to know when investigating the microbial diversity of environmental samples that extracellular DNA increases the species enrichment of these samples by about 55%. So extracellular DNA is here ancient DNA and shows us the historical biodiversity in these environmental samples. So analyzing extracellular DNA here allows us to get some insight into the dynamics and history of the microbiome of environmental samples. In general, natural competence is widely distributed in the environment, and there are a lot of species that can uptake free or bound extracellular DNA. This natural transformation is, of course, a mechanism that is driving evolution and is per se not a threat itself. But of course, with our contribution and our influence in the environment, this can be a threat for the future with the generation of new, more resistant, higher resistant, multi-drug resistance pathogens. Several biotic and abiotic factors influence the release of extracellular DNA, its persistence, and the natural transformation in the environment. As you can see on this figure here, the interactions of all these factors and influences are still not well understood. Therefore, it's of high importance to study the role of extracellular DNA in the spread of antimicrobial resistance genes in different environments. With our project, we hope that we can contribute to increase the knowledge here, also to improve the methods necessary for research in this field. The center of the project are so-called open air labs. We have among the partner countries 3 countries, Estonia, Portugal, and Austria, which have these open air labs. This is an agricultural environment, and in this environment we have several departments, starting with pigs on the farm, pig feces. We have fertilizers, manure from the pig feces or artificial fertilizers. We have the soil from the different fields where the crops are grown. So in this study, we used fields with 2 different crops and also treated with different fertilizers, manure or artificial fertilizers. We have wild animals where hunters were collecting feces in this area. We have the crops that were grown on these fields, and these crops are finally used again as feed for the pigs. But we also have commercial feed that is used as feed for the pigs. Then, of course, we have the people living on these farms. And on the water side, we have drainage water from the fields. We have river water in this area. We have wastewater from the farms, and then also groundwater that is used as drinking water for the farmers and the pigs in these open air labs. To give you a bit more information how these open air labs look like, here the example for Austria. It's located 100 kilometers west of Vienna. The size is about 66 hectares. On the right corner on top, you see a photo of this area, so you can get a better impression how it looks like. We have around 800 millimeter of rainfall in this area. The annual average temperature is around 9.5 degrees Celsius. The main crops grown there are winter wheat and maize, and the dominant soil types are cambisols and planosols, which have a medium to poor infiltration capacity. So over a 1-year growing season, we were collecting from all these different compartments more than 500 samples. In red, you can see here again the 3 countries that collected from all or nearly all these compartments samples. All the other partners were contributing with specific compartments to this sample collection. All the samples were analyzed for antibiotics, pesticides, and heavy metals. From all the samples, extracellular DNA and total DNA was extracted to investigate microbial diversity and also the resistome. Clinically relevant bacterial species, but also some other species, were isolated and cultured from these samples and analyzed using whole genome sequencing, detection of antimicrobial resistance genes out of this genome data. Classical susceptibility testing was performed, and also phylogenetic analysis was performed to get some info about the relatedness of these different isolates that were obtained from these samples. Clostridium difficile was also isolated and used as a model for the transfer from pig to humans. And finally, the idea was to use all this data that were obtained in this project for mathematical modeling, for prediction of risks and risk assessment. Here you can see the first results for antibiotics, herbicides, and elements. In the figure on the top, antibiotics and herbicides in manure samples from 3 countries, Austria, Estonia, and Czech Republic. And we can see some differences here. So the number of antibiotics in Austria is quite low. In Estonia, we have higher amounts of doxycycline, and in Czech Republic, we have even a higher concentration of doxycycline in manure samples. For Austria, and the figure on the bottom, you can see analysis of elements. 8 key elements are shown here. 26 were analyzed for the soil samples, and zinc has the highest concentration in these soil samples here. For Clostridium difficile, the first paper was published this year, and a new toxigenic ribotype 33 was found here, belonging to clade 5 and sequence type 11. And this lineage is known to have the highest zoonotic potential. This clone was found in all the investigated compartments, which suggests that we have a transmission chain here through all the compartments. For the clinically relevant bacteria, the first results from Austria, 89 isolates were obtained. 50% were E. coli, 30% were Enterococcus faecalis, and smaller percentages of Klebsiella pneumoniae and Enterococcus faecium were isolated. In Austria, we couldn't isolate methicillin-resistant Staph aureus and also no Salmonella in the samples. Wildlife and wastewater were the compartments with the highest number of clinically relevant isolates. In general, the diversity was quite high based on classical MLST analysis. Of interest is that E. coli sequence type 1193 was found during this study, which is an emerging clone and which was reported for the first time in Austria last year. 30% of the isolates carried at least one resistance genes, and E. coli is the species with the highest number of antimicrobial resistance genes with 46 genes. If we have a look on the compartments, isolates from drainage samples had the highest number of antimicrobial resistance genes with around 49 genes, followed by feed, river water, wildlife, soil, wastewater, groundwater, and at the end, manure, which was a bit surprising here. 3 of the E. coli isolates were classified as multi-drug resistant. It was also interesting, and I would like to show it here, that during this study we isolated Chronobacter thuringiensis and an outbreak in this wider area where the open air lab is located in Austria was reported among brown hares in this area. In total, 7 hair isolates were obtained. They were genetically unrelated. 5 of these isolates were obtained from dead hares and 2 from hare feces that were collected during our study. Although these hair isolates were not related to each other, but these isolates were related to human isolates. And it's also of interest that all of these isolates carried a new MCR gene, which is conferring colistin resistance. And for those who are interested, it's possible to identify a species depending on the feces. The results on the microbiome performed with 16S amplicon sequencing showed a high alpha diversity with quite similar results for extracellular and total DNA, and except for drainage water and crops where the diversity was low. But in general, on average, 90,000 species-specific reads were found per sample, with a maximum of 1 million species-specific reads per sample. In contrast, the beta diversity was low, except for the soil, diverse soils, and also for extracellular DNA for crops and drainage water for farmers and big barns, where the similarity between these different compartments was quite high. You can see this here on this heat map. On the left side, the results for extracellular DNA, and in these green circles, so you can see that the species similarity is very similar among the different soil types, and also between crop and water and farmers and big barn. A bit different picture for total DNA, where we can only see similar species for the different soil types, not for crop and drainage water and farmers and big barn. So when investigating antimicrobial resistance genes in environmental samples, the standard methods used are quantitative PCR and, in addition, metagenomic analysis. And as shown in this study, PCR has a higher sensitivity compared to standard metagenome analysis. So the recommendation from this study from 2021 is to combine these both methods to really get the best results for detection of antimicrobial resistance genes in environmental samples. When we started with our project, we didn't have the opportunity to use qPCR because all the instruments were blocked for COVID testing. So we were searching for an alternative method, and we ended up with gene enrichment method and with ARGs genetics, which provide at that time the best method for gene enrichment for detection of antimicrobial resistance genes. You can see on top the workflow, so sample collection, DNA extraction, and then gene enrichment capture assay for antimicrobial resistance genes, sequencing, and then the end data analysis. The big advantage of this method from ARGs genetics is that it targets 9,000 antimicrobial resistance markers, which is the most comprehensive database for antimicrobial resistance genes at the moment. Comparing a standard metagenomic assay, which you can see here. So 4 samples were investigated here, and in total, out of these 4 samples, 20 antimicrobial resistance genes could be detected. In comparison, using the gene enrichment approach, more than 100 antimicrobial resistance genes could be detected in the same samples. And comparing also the number of reads between these two approaches, so with a similar number of total reads for both approaches, so for the standard metagenomic analysis and the gene enrichment analysis. In the standard metagenomic analysis, 1.5% of reads were antimicrobial resistance genes, whereas in the gene enrichment, 60% of reads were antimicrobial resistance genes. So we have an increase by around 40-fold here in the number of reads specific for antimicrobial resistance genes, which really increases the sensitivity of this approach for detection of antimicrobial resistance genes. So using this gene enrichment approach from RS Genetics here, the first results from the Austrian samples from the different compartments for extracellular DNA only. The good news is here that the number of antimicrobial resistance genes in river water is quite low also in crops. We were a bit surprised that we found the highest number of antimicrobial resistance genes in wildlife with around 140 different genes, followed by feed, which was also a surprise, by around 80 antimicrobial resistance genes, and manure, which is not that surprising, with 50 antimicrobial resistance genes. If we take a closer look into these different compartments, we can see that there are some interesting differences between the two farms. In the middle, you can see the manure from farm 1 and farm 2, which have quite similar numbers of antimicrobial resistance genes, around 50. But if we go up and if we have a look on the soils that were treated with manure from these farms, the number of resistance genes on farm 1 is nearly double than that of farm 2. And even more surprising is the differences if we have a look on the feed from these two farms. So the number of antimicrobial resistance genes in feed used on farm 1 is nearly 4 times higher than the feed used on farm 2. We still don't have good explanations here, so maybe there is a different antibiotic treatment between the two farms, or if we think about extracellular DNA, of course, the treatment or the usage of antibiotics in the past was much higher on farm 1 than on farm 2. For feed, we actually also don't have an explanation about the differences between these two farms. So, in conclusion, we collected a huge amount of data about extracellular DNA, investigating antimicrobial resistance genes and diversity of species in these samples. Also for total DNA, we collected bacterial isolates. We obtained phenotypic data. We have pesticides, antibiotics, heavy metals from all these sample types, from all these compartments. We have data on the climate, humidity, and also the temperature. And we also used new technology that may also help to improve research in this field in future. And the combination of all this data and all this information may give new insights into the transfer and the development of new resistances in the environment. The project also allowed the detection of new antimicrobial resistance mechanisms in bacterial isolates, and also allowed the detection of emerging resistant microorganisms in the food feed chain and also in wildlife. And it shows also the importance of this somehow neglected compartments in a One Health approach. So finally, we hope that our results can also contribute to the development of strategies as published by ECDC in the technical report on One Health and antimicrobial resistances in 2021. Some additional outcomes, maybe also shortcomings, results where we see that optimization is necessary. So although we thought that we produced a huge amount of data, our mathematicians told us that it's not enough for them to really develop good and accurate mathematical models for prediction of risk and risk assessment. And with this, we also saw that sampling period of a one growing season is not long enough to really have final and good conclusions. From the technical side, we saw that it's really necessary to improve the extraction methods for the diverse sample types. And for future studies in this field, it's absolutely necessary to compare extracellular DNA, and here free extracellular DNA and bound extracellular DNA should be included, and also intracellular DNA must be investigated in one study to really compare the data from all these different DNA sources. For metagenomic analysis to detect antimicrobial resistance genes, the sequencing depth must be increased. So from this standard 6 to 7 gigabyte data output to 2 terabyte, which is really recommended to be sensitive enough when investigating antimicrobial resistance genes in the environment. So with gene enrichment, we think we found a very good tool to increase the sensitivity for detection of antimicrobial resistance genes in the environment. And we are looking forward to use this approach in future projects. Important also to know is that having this many different countries and this many different partners, sample logistics and sending of samples also in these pandemics was really tricky, and it maybe also caused some problems with the data quality at the end. And with this, I would like to thank all the colleagues from the different partner institutions from the different countries who really worked hard to collect all this data, to analyze all this data. And I would also like to thank our external partners, the Open Air Labs, David Weisbrod Group from the Delft University, and of course, ARGs genetics with the main part here, the gene enrichment and 16S amplicon sequencing. Thank you very much for your attention, and I'm happy to take questions.
Thank you, Dr. Ruppitsch, for your informative presentation, your time today, and your important research. We would also like to thank LabRoots and our sponsor, OpGen, for underwriting today's educational webcast. As we will not be answering questions at this time, I would like to continue to encourage you to submit any questions you may have. So before we go, I'd like to thank the audience for joining us today. And any questions that are submitted and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.